Good morning. My name is Bill Skach, and I'm honored to be able to welcome you to the second plenary session. As many of you know, I joined the CF Foundation's medical team in the national headquarters in July of this year, and I'm absolutely delighted to be able to work among these outstanding individuals for this exciting purpose. Before we begin today's presentation, we have one very important task. And for this, I would like to introduce a long-standing and respected colleague, Chris Penland. Chris is the Vice President of Research at the Foundation, and for the past 15 years has been skillfully guiding the basic science and the preclinical drug development efforts at the Foundation. This means that most of the posters and scientific abstracts and presentations here have somehow had Chris's fingerprint on them. So I'm truly delighted to introduce him. He will be presenting the Richard C. Talamo Award, which is one of the highest honors given by the Foundation for outstanding contributions in CF care. So join me in welcoming Chris to the stage.
Thank you, Bill. So we are gathered here in Atlanta to review and communicate the most recent insights into CF pathogenesis and care. The tremendous advances that we are learning of, however, do not exist in a vacuum and are based upon the work and commitment of many devoted and talented people who helped pave the way for our current success. Today, we remember one of these past leaders, Dr. Richard Talamo, and present 4 individuals the prestigious award named for him. Dr. Richard Talamo graduated magna cum laude from Harvard College and earned his medical degree from Boston University. He pursued a career in pediatrics and became an NIH clinical fellow in the laboratory of Dr. Paul de Saint-Ignace, where he developed what was to become a lifelong passion for understanding CF and caring for those with the disease. While a professor of pediatrics at Johns Hopkins, Dr. Talamo became deeply committed to the mission of the CF Foundation and served in many capacities, including on the Medical Advisory Committee and Board of Trustees. During this time, he helped raise interest in CF and encouraged qualified researchers to focus on the disease. Dr. Talamo returned to Boston in 1980 as the chairman of pediatrics at Tufts University. Sadly, this brilliant, productive, and caring man died prematurely at the age of 47. The Richard C. Talamo Distinguished Clinical Achievement Award recognizes individuals who have spent their careers researching and caring for people with CF and whose contributions have significantly impacted this disease. Now, today's ceremony has actually been 40 years in the making. So before we welcome the awardees to the stage, we should recall that long before the CFTR gene was identified 25 years ago, we knew that Airway's disease contributed greatly to CF morbidity and mortality, and that routine respiratory therapy, including saline-aided sputum induction, was a vital component of treatment. Hypertonic saline's potential as a means to improve mucociliary clearance from the lungs was recognized in the late 1970s in chronic bronchitis. Peter Middleton and colleagues at the Royal Brompton Hospital first demonstrated its potential in CF when they described a saline-dependent increase in nasal mucociliary clearance. In 1996, a 2-week open-label study of hypertonic saline led by Colin Robertson extended this work by demonstrating a significant pulmonary benefit in CF patients. Later that same year, Michael Robinson and Peter Bai in Australia reported an improvement in lung mucociliary clearance in CF patients following an acute hypertonic saline challenge, and they quickly followed that with a study demonstrating augmentation of mucociliary clearance was dose-dependent. Peter Wills and colleagues in London subsequently hypothesized that the mechanism of salinine's beneficial effect on mucus transport was due to osmotically sensitive changes in mucus rheology, a hypothesis that was later confirmed by Robert Tarrin. This relatively inexpensive approach to airway clearance was further developed over subsequent years by a number of investigative teams across the globe seeking optimal combinations of delivery devices, saline concentrations, breathing patterns, outcome measures, and patient populations. 2006 saw this work come to a head with back-to-back papers in the New England Journal of Medicine. In these papers, hypertonic saline was found to be beneficial and safe in a large population of CF patients, yielding a durable mucus clearance effect. This work led to the widespread uptake and use of hypertonic saline as a chronic therapy in CF. It has also laid the foundation for subsequent studies seeking to understand the potential of hypertonic saline in young children with CF and opened the door for further studies into the interplay between mucus, ion transport, water, and CFTR function. Please join me in recognizing the efforts of Drs. Mark Elkins and Peter Bai, who conceived of and conducted the Australian study, Dr. William Bennett, who has strived to improve the in vivo measurement of lung function, lung mucus clearance, and Dr. Scott Donaldson, who conducted the complex U.S. study and has since worked to bring forth mucociliary clearance as a clinical trial outcome measure in CF. Peter and Scott, if you can come forward. Unfortunately, I don't think Dr. Elkins or Bennett were able to come to the meeting, but we will surely send their awards on to them later. Peter?
Thanks very much.
Just come out in 2 pieces. Yeah, and use it later.
Thanks very much. I really love coming here. This is almost as big as my footy team winning the premiership this year, South Sydney.
Peter, just wait right there. Scott?
Well, just a very quick thank you. First of all, I'm glad I showed up this morning. I was busy practicing my talk for the last 5 minutes, and they're talking about hypertonic saline. Okay. But thank you. I appreciate it.
Thanks to Bob and the US Foundation for giving us all the funding and incredible support over the years.
Thank you, Chris. Today's plenary session will be presented by John LaPuma, who many of you know for his outstanding work in contributions to the field of CF microbiology and infectious disease. John is currently the James L. Wilson Professor of Pediatrics and Communicable Diseases and the Division Director for the Pediatric Research Infectious Diseases at the University of Michigan Medical School. Over his 35-year career, John has been the consummate physician scientist. He's published over 150 papers dealing with cutting-edge molecular diagnostics of bacterial strains and species. He's identified dozens of novel bacterial species and he's developed new methods for culturing microbes and animal models for understanding how infections in the CF lung take place. But that's the easy part, because that's just the science, John. What's really amazing is that he's been able to take these concepts from the lab and the bench directly into the clinic, and it's had a major impact on how we think about CF disease in patients and individuals, and also how we can control and treat that disease. John has made particularly important contributions to our understanding of B. cepacia, how it's transmitted, what its complications are, and he's been very involved in the development of the infection prevention and control guidelines that are used to protect individuals with CF today. So what does this really mean? Well, John's lab has a repository of 35,000 bacterial strains, and this serves as a national repository for the CF Foundation. So if a sputum sample is sent from Atlanta and it goes to John's lab, he can immediately determine if that same bacterial strain infected someone in Seattle or Denver or Baltimore. And this is incredibly important for tracking how infections spread among the CF community, how effective we are at eradicating those infections, and really understanding a lot of this transmission process. So this is really an incredible role and an incredible important work that John has performed over the years. In addition, he's played very important roles for advisory roles for the CF Foundation in the Therapeutics Development Program, in the Antimicrobial Task Force, the Data Safety Management Board, and many, many others. Today, John is going to take us on a historical tour of CF, describing where we've come from, where we are, and where we're going in infectious disease in the CF world. The title of his talk is CF Microbiology, Past, Present, Future. Please join me in welcoming John to the podium for what I'm sure will be a fascinating discussion.
Thanks, Bill. Thank you. Thank you, Bill. And thank all of you for coming this morning. And thank all of you who are out there joining us remotely. Good morning. Welcome. No disclosures. I'll start with a few acknowledgments that I think are very important. First of all, the CF Foundation, without whose support over the years I wouldn't be standing here this morning in front of you. Secondly, the conference program committee for asking me to do this talk this morning. It's really a great honor, and the program committee's conscription of me was persuasive. I'd like to also thank Dutch van de Vender, who was really very helpful in developing not only the content, but also the slides for this presentation. So if there are any slides that you find anything less than appealing, I know Dutch would like that feedback later. So he'll be out in the hall afterwards. And a couple disclaimers to start with. I really wish that I could mention everyone who's been involved, the scientists, physicians, sponsors, who've contributed to our understanding of CF microbiology over the past few decades, but of course in the time I'm allotted this morning, that's really not possible. This is another way to say I'm going to drop a lot of names, and I'm really sorry if yours isn't one of them, but please forgive me. Also, I want to make clear that this presentation is not intended to represent a consensus statement from everyone involved in microbiology. I'm going to tell you the story of CF microbiology as I see it. I think you could have other people come up who would tell you the story in a slightly different way. So there is no secret society of CF microbiologists that I'm representing. This is the world according to Lipuma this morning. Now, that said, I did have a bit of a strategy for developing this talk. When I was asked to talk about microbiology, it's a very broad topic, and I thought I should reach out to some colleagues, some trusted friends. And so I asked people, and I'll show you the list at the end of the talk, and I said, please try to give me, in just a few bullet points, tell me what you consider the most important developments in CF microbiology in each of the three categories here. And these friends of mine complied and sent me some really interesting information. And what became very apparent when I got that feedback was that microbiology really means different things to different people. And people's heads go to different places when they think about microbiology. Right, so some people think about clinical microbiology, detection, identification of microbes, taxonomy, nomenclature, susceptibility testing, all those things that are done in your clinical microbiology laboratories locally. Some people think more about infectious diseases, diagnosis, treatment, pathophysiology of infection, outcomes of infection. Still others really look at basic microbial physiology and virulence, really understanding of pathogenic mechanisms, host-microbe interactions. And still others, when they think about microbiology, they think infection control, epidemiology of infection, ecology of microorganisms, impact of the infection control guidelines that we have now. And so it really is a bit of a mixed bag, and I think I'll be touching on all of these themes in the next 90 minutes? No, 40 minutes. The other thing that was interesting was that even past, present, and future seemed a little bit dependent on the person who was considering that. For example, among the people I asked, at least one person said that inhaled antibiotics were one of the most important past developments. Someone else said it's really one of the most important present, and yet another person said inhaled antibiotics are one of the most important future. So what's clear is that there really aren't these clear demarcations between past, present, and future. CF microbiology and the evolution of CF microbiology has occurred on a continuum, characterized more by incremental advances than by big quantum leaps. So over the next little while, what I'll attempt to do is describe this evolution, and part of this will be sort of a history of early CF microbiology, so I'll do some review of what things looked like when we first started thinking about microbiology and CF, where that led us for the past 30 years or so, and where I think we're headed with CF microbiology. So where do we start? Well, most people, when they're talking about CF, start with Dorothy Anderson, describing CF in 1938. But of course, CF was with us long before 1938, and when it was described by Dorothy Anderson in 1938, it was within the context of what we knew about microbiology at that time. So it's worth a little bit of a review of microbiology. So we'll start with a very brief history of microbiology. Where do we begin with that? Well, before 1920, I can tell you. 1546, maybe, germ theory, Girolamo Fracastoro. Not a name familiar to a lot of you, but germ theory was described long ago. Most people talk about starting with von Leeuwenhoek, looking through his primitive microscope, seeing little things swimming around that he called animalcules. But really, we can leap forward a bit to the latter half of the 1800s, what's called the golden era of microbiology. And you have these giants of microbiology. Louis Pasteur, who came up with some of the early ideas surrounding vaccination and ways of processing food, a process we now call pasteurization, which essentially heat shocking microbes in foods to decrease their density. Robert Koch, Koch's postulates. This was very important. You remember that what Koch did was he took a microorganism from a diseased animal in order to prove that that was the thing that was causing the disease, put that organism into a healthy animal, got the same disease, therefore fulfilling Koch's postulates that one organism can cause one disease. This is a paradigm that has dominated infectious diseases for many, many years. And then Graham and Petrie work with culturing, identifying organisms in the laboratory. So again, very important people, the golden age. Some people have referred to this as the great bearded age. But these are the giants. So let's now sort of leap forward 50 years and Alexander Fleming and the discovery of penicillin. And we all know the story here from our high school biology classes. Fleming was working with Staph aureus, and he had a bunch of cultures, and he had them on his desk, and he went away on vacation. When he came back from vacation, he found that something had been contaminating his cultures and killing the staph that was on those plates. Turns out that that was a mold, a penicillium mold, and from there we have the discovery of penicillin. As an aside, it's never been really entirely clear where the source of the staph that Sir Fleming was using at the time. I think that this slide gives a little bit of a hint, particularly if one looks at the position of this bacterial loop that he's using. It seems to suggest that, at least I think we can speculate as to where that staph may have been coming, but that's speculation. I don't know. Okay. All right, so moving to CF microbiology then. All right, so I'm going to break this down into what I consider sort of three phases. The first phase is the Pre-Pseudomonas Era. So we have Fleming here in 1928. Penicillin really wasn't commercially available for some years later, 1942. The sulfonamides actually came first. And it was within this context of the early development of antibiotics that Dorothy Anderson described CF. And then Sidney Farber came along in 1943, and Sidney Farber said, well, respiratory tract damage in CF depends on primary obstruction by thick mucus, failure of proper lubrication of epithelium, and secondary staph infection. So now there's this connection between staphylococcal infection and the bad effects of CF. Streptomycin came along in 1945, and then the year later, 1946, Paul de Saint-Denis talked about using inhaled penicillin, really kind of a radical idea at the time to treat CF. And it was based on this work, and here's a paper from 1946 with Dorothy Anderson. And he's looking at post-mortem cultures, post-mortem from lung, and look at the ages of the children here. All right, this is in months. So the oldest individual here is 40 months old, and what you can see is that Staph aureus really was the dominant organism found in post-mortem cultures of lungs. So this was the dominant pathogen responsible for death, early death, people with CF. Well, a few more antibiotics were developed then in the 50s, and this became known eventually as the antibiotic era. Rapid development of new antibiotics, and you can see some of these antibiotics from the 1950s. A couple of them really still very important in CF, colistin, vancomycin. And then around here, 1960 is when Harry Schwachman said, well, you know, we ought to be using all of these things. Tetracycline, combined with erythromycin, and a sulfa drug. And let's aerosolize penicillin like Paul de Saint-Denis had told us. And maybe throw in streptomycin, neomycin, polymyxin. Let's throw all these new antibiotics at people with CF to see if that makes a difference in prolonging their life and in curing this staph infection that seems to be the cause of death. A few more important antibiotics then in the 1960s. So I just want to give you a timeline of when these antibiotics were developed. We tend to think these antibiotics have been around forever, but you'll see that many of them are actually rather recent. And what we saw, coincident with the development and use of these antibiotics, is remarkable change in CF. So if you look here, this is diagnosis or survival from the time of diagnosis. So in the 5-year period, 1952 to 1957, about 30% of children survived 2 years after diagnosis. In the next 5-year period, 57 to 62, now we're looking at maybe 80% of children survived 2 years after diagnosis. In the following 3 years, 62 to 67, well, now look, it's maybe 80% are living 5 years after diagnosis. So a remarkable change, coincident with the development and use of antibiotics. Now, I don't want to imply that antibiotics were the only part of the story. I think there are a few nutritionists out in the audience who might take issue with that. So there was a lot going on. But antibiotics, I think, was a very, very important part of this story and this early success in CF. Now, the other thing that happened around the time of all these antibiotics being developed was that we developed antibiotic susceptibility testing, another concept that I think people think has been there and with us forever. Really, it's not that old. This is 1966. This is the what we now know as Kirby-Bauer susceptibility testing, in which we take an organism that we pluck from a colony from a culture, that is, and we plop it on a plate, and we look at the zone of inhibition, which indicates the degree of susceptibility. So here is this plate, and what we've done is we've taken a colony and we've spread it all over this plate, and then we plop down these little pieces of paper that have known concentrations of antibiotic in them, We put this in the incubator overnight. After incubation, we then see what these zones of inhibition are, and we can measure this zone size. So here is an antibiotic to which this bacteria is quite susceptible. Here's an antibiotic that doesn't seem to bother this organism, grows right up to it, right? But remember, this is on a dish, right? This is Petri. And so how do we then translate what we're seeing on a dish into something clinically useful? Well, there were a couple of committees developed, then one in the US and one in the UK, that took this testing and used it to develop criteria to determine susceptible and resistant. And what this really means is the development of susceptibility breakpoints. And this is really, these are antibiotic levels that predict treatment response. And what this did in the early 70s, this kind of novel idea of susceptibility testing, was that it now changed the way we were using antibiotics. So we're now using antibiotics, we're choosing them based on the susceptibility testing. We're not just throwing them at people like Schwachman had done with great success. We're now thinking we ought to look to see what the susceptibility is in vitro, use antibiotics in that way. And what this did was it raised some concern about resistance. And this concept of, well, maybe we ought to be saving antibiotics. We don't want to use them up too quickly and have the organisms in an individual become resistant. Maybe we ought to be a little more judicious and think about saving some things until we really need them when the person's really sick. Well, we saw a few more antibiotics then developed in the 70s, and again, some very important ones, amikacin, tobramycin among them. And what we saw again was a dramatic increase. Now here's predicted median survival in the 15-year period between 65 and 80, really doubling from 11 years to 22 years. Again, coincident with the development and use of new antibiotics and the aggressive use of antibiotics. So I would summarize this pre-Pseudomonas era in this way: the identification of CF, the recognition of the importance of infection in CF, the development and use of lots of new antibiotics, and increased survival, dramatically increased survival. So what this did in this period was it fundamentally changed CF. CF is no longer a disease that kills children in the first few years of life. We now have extended life. We now have adolescence with CF, something that we didn't have previously, something new on the planet, in a sense. And in response to having now adolescents with CF, we needed to develop social programs. We recognized the importance of psychosocial needs, summer camps, social programs. And we had this emerging concern about resistance. And what's going to happen if we have these adolescents who now have bacteria that are resistant to lots of antibiotics? And again, maybe we ought to be parsing those out a little more and saving them until people really need them down the road. Well, this leads to my next era, which I'll call the Pseudomonas era. A shift in CF microbiology. Now Pseudomonas is the primary opportunist. And what that did was it encouraged a large number of studies looking at the epidemiology and the associated outcomes of CF. So a number of studies with over 5,000 individuals studied in the 90s, early 90s in particular. And we learned a number of interesting things with these outcome studies and these associations. For example, one of the things we found was the impact of mucoid Pseudomonas and this phenotype on survival. Here's a study, an 8-year study by Henry and colleagues. And what was seen was that with no Pseudomonas, this is a survival curve, so cumulative survival. With non-mucoid strains, you see some decrease in survival over years, but with this mucoid phenotype, really a dramatic difference in survival. And this hadn't really been known until that point. So again, this isn't that long ago that we made this realization. Now, the other thing that happened in this time period is that molecular biology happened. And by that I mean there was this explosion in our ability to understand and to use tools of molecular biology. Starting in 1970 with the development of restriction enzymes. These are very important, fundamental to our ability to clone DNA from bacteria. DNA sequencing in 1977. The development of polymerase chain reaction, which really revolutionized revolutionized our ability to manipulate DNA in '83. The first automated sequencer in '87. We know about CFTR in '89. And then sequencing of the human genome and Pseudomonas in 2000. And just to put this into context, what we call now next generation sequencing in 2005. So we had sort of a convergence of events. All right, we had older individuals. Now with Pseudomonas, we're learning a lot about Pseudomonas epidemiology and outcomes. We're learning about these associations with some unusual phenotypes. And we have now all these molecular biology tools at our disposal. And what that did, having those tools, allowed us now to understand some of the molecular mechanisms behind some of these interesting observations we were making. For example, here. So here is a Pseudomonas cell. Here's the outside of the cell up here. And we learned about this phenomenon of mucoidy. And what we now know is that this protein, MUC-A, which normally binds this sigma factor, which is a part of the RNA polymerase, isn't able to bind it when it's mutated, which happens under stressful conditions, such as in the lung. And when it liberates this sigma factor is then able to do other things. It can turn off production of flagella and pili, type 3 secretion, so it downregulates motility and virulence, and it upregulates the ability of Pseudomonas to exist in hostile environments, these stress responses, and this alginate production. All right, so we worked out this mechanism that we had observed phenotypically some years prior. The other thing it did was it allowed us to understand a great deal about the basic biology of Pseudomonas. So, for example, Pseudomonas quorum sensing. These are very important systems in bacteria. They are systems by which bacteria can sense and respond to neighboring bacteria, so-called bacterial crosstalk. And then these are systems that are also used by bacteria to coordinate gene expression based on local density. We know that Pseudomonas has a number of quorum sensing systems, 3. A couple of them produce these homoserine lactones, another produces a quinolone, and when these compounds are leached into the environment around a Pseudomonas strain, they are taken up by other Pseudomonas strains, and these other transcriptional regulators are upregulated to produce virulence factors, biofilm formation. We learned a lot about virulence and host interaction. This is from a beautiful review by Alan Hauser and Egon Ozer. It's published a few years ago. And I apologize for the utter simplicity of this slide. For those of you who study, so I mean, this is like 30 years worth of work here, very briefly summarized, and I don't want to spend a lot of time with this, but we learned an awful lot about how Pseudomonas, so here's Pseudomonas here, sitting on top of an epithelial cell. And we know that Pseudomonas can bind to the epithelial cell in a number of ways, by flagella, by these type 4 pili, hooking onto this acyloGM1, which is essentially a combination of fat and sugar. This lipopolysaccharide can interact with these toll-like receptors to upregulate pro-inflammatory cytokines. We see this type 3 secretion system I mentioned a few minutes ago that can pump these toxins into the cell. And these toxins do all kinds of horrible things. They disrupt the normal regulation of the cell. They destroy the cell's integrity by attacking this cytoskeleton that the cells have. So we learned an awful lot about all of these mechanisms. And this is, again, work that has taken years and years to do. Alice Prince and Jerry Peer and a number of other people, Steve Laurie, I could go on, and a number of people who've contributed to this work, a large body of work getting us to understand how all this stuff comes together. Talked about biofilm formation, very important, right? Pseudomonas are able to form these nice biofilms. They can attach to the surface. Here's a cell, again, an epithelial cell. They form these biofilms that are these little collections that protect bacteria within them. We know a lot about how these biofilms are made. We know the composition. They're made of polysaccharides, DNA from cells that are lysed, this alginate that we talked about, so we know a lot about the composition. We also know about how biofilm function. Biofilm functions by producing this really protected niche, this region that has a gradient of oxygen and nutrients, so high on the outside, lower on the inside. The cells on the inside are rather metabolically inactive, kind of quiescent. And these biofilms then produce a barrier to antibiotics. They upregulate, and so this is another reason why we see different levels of antibiotic resistance. The cells closest to the outside of the biofilm see a little bit of antibiotic, upregulate their antibiotic resistance mechanisms, whereas the cells more towards the center don't see the antibiotic and don't become resistant. We also know that there are these rhamnolipids produced that degrade neutrophils. So again, we've learned an awful lot about these mechanisms. Well, along the same time we're learning all that about Pseudomonas, this antibiotic era extends now through the 80s and 90s. A further explosion of the available antibiotics we have to treat CF. And again, you can see some very important ones here. Azithromycin, aztreonam, meropenem, levo. These are all in the 1990s. They haven't been on the planet since, they're new, right? So I want to, again, try to put these into context. We also know that, as I said, inhaled penicillin was being used by de Saint-Ignace many years ago. The availability of these newer agents then stimulated more formal trials using other antimicrobials in inhaled format. And so you can see some of these here. And what this enabled, the development of all these antibiotics, the use of inhaled antibiotics, then stimulated a large number of studies using antibiotics now directed against Pseudomonas. And again, you can see these here, nearly 2,000 individuals studied, and this includes the pivotal studies of tobramycin and aztreonam, which then led to the approval, finally, of Toby and Caistin, which in turn led to our current treatment strategy for using these two agents, which is shown here. Now, the other thing that happened around this time is that we learned a lot about interpatient spread of microorganisms in CF. Now, we knew that Burkholderia from the late 80s and 90s seemed to be able to spread between individuals with CF, particularly some strains of Burkholderia. We didn't think that that happened a whole lot with Pseudomonas until this paper. All right, this is in 1995. This is from the Liverpool CF Center. And what was found there was that there seemed to be an increasing incidence of ceftazidime-resistant Pseudomonas. So this was found in 65 of 92 children who were infected with Pseudomonas. And some of these children had never been treated with ceftazidime, so this seemed rather odd. Typically, you see ceftazidime resistance after one initiates treatment with ceftazidime, but these are children never treated, and now they have this ceftazidime-resistant strain. When that organism was genotyped, it was found to be the same amongst all of these, or most of these. 55 out of 65 had the same strain. This has subsequently been called the Liverpool epidemic strain. This Liverpool epidemic strain of Pseudomonas was subsequently found to be in about 48% of clinics in the UK. About 11% of people with CF in the UK were infected with the same strain. And the really more important part of this is that that strain was also associated with a poorer outcome. This same strain was then found not too long ago by Sean Aaron and colleagues in Ontario. About 15% of patients in Ontario had this strain, and again, the same observation, increased rates of death and transplantation. Unknown whether this strain is found in the US. We really haven't looked very hard in the US. We haven't done the sort of surveillance type work that needs to be done. In addition to this strain, this Liverpool epidemic strain, a number of other epidemic strains have been identified in various countries. Again, mainly found just by surveillance, not really investigating the outbreaks, but surveillance, just ongoing, looking, genotyping. And what is found is that when you look for these strains, generally you find them. There's really only one that's been found or described so far in the US. Early work in my lab suggests that we will find common strains of Pseudomonas aeruginosa in the US as we do more large-scale studies. The other thing that happened around this time was we now started to question the utility of that susceptibility testing that I talked about a few slides ago. So this is a really interesting study from Juliette Fauriker, and what Juliette did was, let me walk you through this, she took 48 colonies from the same sputum sample, 48 Pseudomonas. And she tested these over and over with this Kirby Bauer, and then measured these zone sizes that we talked about for different antibiotics. And each of these lines represents one or more colonies. And so what you can see, for example, here, if we look at meropenem, here is one colony from the same culture, one colony that has a zone size of around less than 10, that would be resistant. Other colonies, 2, 3, 4, all the way up to a zone size of 50, meaning very, very susceptible. So there seems to be a lot of variability in this sort of susceptibility testing. And then there were a couple other studies that came out that really showed that susceptibility testing doesn't predict response to antibiotic therapy very well in CF. So to sort of summarize this Pseudomonas era, we had this increased survival from the previous era. Molecular biology came along, enabled a number of really important studies that opened our eyes about the basic biology of Pseudomonas. We did a lot of studies looking at Pseudomonas outcomes. We developed more antibiotics that led to trials of using these antibiotics to treat specifically Pseudomonas. We realized there were Pseudomonas epidemic strains that were out there. And there started to be this questioning, this bit of a disconnect between susceptibility testing and outcome. And what this led to then was intensified infection control, a little bit less concern about resistance. We weren't quite sure now really what that means. And it really led to our current strategies of treatment, and that is eradication, early aggressive treatment that is, suppression of chronic infection, and episodic treatment for exacerbation. And these are strategies we still currently use. So the next era then, the third era, is the way I see it, is the Pseudomonas 2.0 era. Now here, I just want to go back a bit. This is a paper that was written by my friend Peter Gilligan and was really the standard work for 2 decades on the microbiology of CF. This was written in 1991. What Peter said in 1991 was that strep pneumoniae, a few other enteric bacilli, gram-negatives, other than aeruginosa and what was called Pseudomonas cepacia at the time, are occasionally recovered. But like H. influenzae, none of these really seem to persist for any extended period of time. And it seems that any role they have in causing disease is likely to be secondary to that of aeruginosa and other organisms. So this was the paradigm. This is what we thought in 91. And the reason for that was when we look at the gram-negatives that we understood, that we were able to culture from people with CF, in the 1960s, it was Pseudomonas, it was H. influenzae. In the 1970s, occasionally we saw Stenotrophomonas, Ralstonia. In the 80s, rather, a few more Pseudomonas, non-aeruginoses we would see occasionally. We started to see a Chromobacter, Burkholderia, and this is when Peter is writing his paper. There isn't a big number of organisms that we're culturing from people with CF. Of course, in the 80s, then the Burkholderia cepacia became the Burkholderia cepacia complex. We started to see B. gladioli, not a member of the complex, but a common Burkholderia in CF. Yeah, in 1990s, we start to see now even more things, this whole new genus called Pandaria, Inqualinus lamosus, an entirely new genus that we found primarily in people with CF. Now we're up to 18 species in the B. cepacia complex. There will soon be more. Ralstonia exploded, Cupriavitis exploded, so you can see where we're going here. Yeah, we're gonna need a bigger slide. Yeah, so, and now, moving along, now a lot of Chromobacter. So this just keeps expanding, right? We're gonna really explode pretty soon. Now the other really interesting thing that happened around this time was there's this brilliant paper that I absolutely still love. This was a seminal paper. This is work by Ken Bruce at King's College in London with Geraint Rogers. And this is really sort of prescient. This was published back in 2003. This is before the Human Microbiome Project started. And what Ken did was he used culture-independent methods to assess the microbiology of airways in CF. And what was found, so what this does, right, it allows us to take specimens, extract the DNA from those specimens, analyze that DNA to tell us what microbes are in that specimen without the need to culture them. And there are a number of ways to do this. We currently now use next-generation sequencing, and we are now familiar with the terms microbiome. These are all the genes and the gene complement of all the microbes in a given sample, or metagenome, which are not only the microbes, but all the host, all the genes in a particular sample. So we use microbiome and metagenome and microbiota. These are terms that are now becoming familiar to us. And what we found, and Ken found, and others have found subsequently, is that the airways seem to be the home to very diverse microbial communities. This is from a paper we published a few years ago, and let me walk you through it. This is one patient, 30 sputum samples collected over 8 years. Here are the age, this is the age of the person, and the dot is the age at which the sample, each of these samples was collected. And each line here represents a sample, and each color is a different species within that sample, and the size of the color tells you the relative abundance of that species within this community. And what you can see here is that we see quite diverse species. Green here is Pseudomonas, the yellow is staph. If we were to get a report of these cultures back from the clinical micro lab, we would see this. The species included in the report, staph and Pseudomonas, are in blue. The things in the organisms, the species in this orange, maybe it's maize, because it's maize in blue. Maybe this is a maize color. These are, it's an inside. These would not be in the culture report. So the point is we seem to be missing from the microlaboratory a number of species that seem to be present in fairly high abundance. Now there's questions about whether these species are all upper airway, whether they're really in the lower airways or not. This is a paper I want to highlight. This just came out last month from Luke Hoffman. And what Luke and colleagues did in Seattle was they had a 3-year-old with CF who had very bad disease, had an excised right lower lobe. And they took this lobe and they partitioned it. And they did this very sterilely, very carefully. And then they did deep sequencing on these different sections. And what they found was, in fact, lots of microorganisms in lung. So these now are not from upper airway. These were specimens that weren't contaminated by expectoration. And what they found was H. influenzae, Ralstonia, a number of anaerobes, including Propionibacterium, in fairly large numbers. This is, again, relative abundance. These are the samples from each of these different segments. And these were very poorly reflected in the oropharyngeal swab from that individual. So what this tells us is that there are diverse communities in the lower airways. Oropharyngeal swab may not really reflect very well what's in lower airway, and severe disease can occur in the absence of those traditional pathogens. There was no Pseudomonas here. There's very little staph here. So I think this is a very important observation. It's an N of 1, it's a small study, but I think it illustrates a very important point, and I don't have time to discuss lots of other literature that suggests that what we're seeing with deep sequencing of expectorated samples really does reflect, in large part, what's in lower airways. Now, the other thing that's really important to understand is, in addition to having lots of things in lower airways, the organisms that are in lower airways, in fact, organisms that are next to each other in any niche, are interacting with one another. Organisms are not inert to one another. They're not agnostic of each other. They're always interacting. This is a study from Chris Sibley and Mike Surratt. This is when Mike was in Calgary. He's now in Hamilton at McMaster. And they did a clever study. They used Drosophila. They used the fruit fly as an in vivo model. They actually did this with mice as well. But what they did here was they took Pseudomonas aeruginosa alone and infected the fly, or what they called oropharyngeal flora, normal flora, or both. And what they found was depicted here. Here are the number of live flies over days post-infection. So if you infect with only the oropharyngeal flora, you don't see a lot of death, right? No bacteria. In fact, in some ways it looked like those that got the oral flora did better. Strangely, if you infect with Pseudomonas, you see death. But if you infect with both, you see much greater death. So there seems to be some interaction. And they worked this out pretty well. What they looked at were a number of genes that seem to be involved with virulence. And they saw here, it's a little hard to read, but let me just show you what they found, was when there were both Pseudomonas and this other species infected together, they saw upregulation of a number of genes beyond what they saw with Pseudomonas alone. And down here, these quorum sensing related LAS I and LAS B, these are transcriptional regulators. Here's Pseudomonas alone. In Pseudomonas plus another species, you see upregulation of Pseudomonas activators that might increase virulence. Now, there are a number of other studies, and I wish I had time to go through this, that show us time and time and time again interactions between microbes when they are in close proximity. And I only have time to show you a couple. Here's one of my favorites, still one of my favorites after a number of years. This is from Luke Hoffman when he was in Sam Miller's lab in Seattle. And what Luke found was that when you grow Staph aureus in the presence of Pseudomonas aeruginosa, Pseudomonas produces a compound called HQNO, and HQNO, when Pseudomonas and Staph are together for days, and particularly if you add aminoglycosides to that mix, what you end up with are small colony variant Staph that are much more resistant to antibiotics, including the aminoglycosides, very difficult to treat. And they found the presence of HQNO in CF sputum, suggesting that this mechanism of Pseudomonas doing something interesting to Staph is occurring in vivo. Here's another illustration of this story. This is by Marv Whiteley and colleagues in Texas. This is just beautiful work, and they've done a number of papers I wish I had time to tell you about. But what they find is that peptidoglycan, which is a structural component of both gram-negative and gram-positive bacteria, is a sort of universal sensor. Other bacteria sense the peptidoglycan from one species. Eukaryotic cells, human cells, sense peptidoglycan. And in the presence of peptidoglycan from gram-positives and gram-negatives, Pseudomonas increases a number of virulence factors. So it's felt to be that Pseudomonas has this way of sensing what's around and responding to that. So the behavior of Pseudomonas in the presence of other things and in the presence, depending on the niche they're in, will behave differently. So we can really hope that a lot of what we found with Pseudomonas and the brilliant work done that I described earlier really still pertains to the CF lung and to the different microbes that we find connecting with Pseudomonas. So these communities are diverse. They're likely interacting with each other in ways that then change the behavior of the individual species when in isolation. Let me go back. Okay. Now, if that weren't enough, it's even more complicated. So there's even diversity not only within this community, but there's diversity within species. So this is from Pradeep Singh, and Pradeep did a clever experiment where he took CF sputum and he cultured it, and he took a number of Pseudomonas aeruginosa, and then he takes individual colonies and he assesses phenotypes of those individual colonies from this plate. And you can assess these for a number of phenotypes that seem to be important in virulence. And what he found was that, again, from one sputum culture, one individual, different colonies, you can see that colonies have different nutritional requirements. So here's a plate now. Here's one colony that's able to grow on this plate, another colony that can't grow without certain amino acids. Swimming motility, now this is in a little well, and you can see that here's a cloudy bit of media because the bacteria are swimming around, but these bacteria can't swim, they sink to the bottom. Here's antibiotic susceptibility, so here's a plate that contains Cipro. Here's one colony from the same individual, same time, same specimen that's totally resistant, and here's one that isn't so resistant. It sort of explains what Juliet found a few years ago. Now, another interesting thing is that there are a number of these morphotypes. So this is a study that has not yet been published. Vaughn Cooper and University of New Hampshire shared this with me. Here's a Pseudomonas aeruginosa colony. You grow this in a biofilm. And then over a number of days, 17 days, 45 days, 90 days, you get this amazing array of different morphotypes. Really striking differences. And what's interesting about this is that this, I think, we believe, has some real biologic meaning. This isn't just a clever parlor trick that you can impress people with. This is meaningful. And this is something that Soren Mullen and Niels Hoiby and others have done. They've proposed this mechanism by which in an individual, the initial infection with Pseudomonas is by sort of garden variety Pseudomonas. And maybe that can be cleared in lung initially with antibiotics, and because the lung is a fairly hostile environment, the sinuses are a much less hostile environment, so Pseudomonas can persist there. And as they persist, they start to adapt into these different morphotypes, just like we saw with that in vitro experiment that I just showed you. And as these adapt and become sort of accustomed to this niche, they're now much better at infecting lung. So maybe the adaptation occurs here up in the upper airways and then is the source for these sort of pre-adapted or now adapted strains that have all these different morphologies, different functions to exist in the lower airways. So we've got layers of complexity. We've got diverse microbial communities, dozens of species, variants within individual species, maybe many dozen, spatially distinct. We showed you Luke Hoffman's paper where there were different species in different parts of lung. So one germ, one disease, not so much. The communities and variants within these species are interacting with each other and with the host in ways that change the species and the community in ways that further change. This is a very dynamic process. The microbes are changing each other, they're changing the host. That change in the host stimulates more change in these species. All right, so how do we understand this complexity? How can we begin to possibly understand this very complicated business? Well, we take a systems biology approach. And systems biology is defined here a few years ago. So rather than a reductionist view, it's really the pluralism of the causes and effects and networks that we observe by looking at multiple components simultaneously. And there are ways to do this. Here's some unpublished data from my lab. Looks very confusing, but what we're able to do now is draw these co-occurrence, these networks. And what we see here is that the size of this node is the number of co-occurrences. These are species that we find together. The thickness of these lines tells us the strength of the positive interaction, the positive correlation. The red lines are negative correlations, so we don't see Pseudomonas. For example, with lots of things, Pseudomonas tends to come in and crowd other things out. But you can develop these very clever networks. This is one I don't expect you to see the words, but the point is that you can use these. This is work from Gisli Eisenson and Michael Tunney, Stuart Elborn in Belfast. And what they're doing is you can use these network sort of analyses to look at different parts of the airway. You can look at different maybe time periods when someone's having an exacerbation versus when they're stable. So we can start to tease apart what these complex webs of bacteria look like. You can also, what we've done, also from my lab unpublished, but we can look at these, I showed you this before, this is again, each bar is a separate sample. What we've done is done deep sequencing on consecutive days around the time of an exacerbation. So here's when this individual is completely without symptoms. Symptoms begin here. They start to have some symptoms of exacerbation that then culminate with the start of antibiotics. And you can see how these communities seem to be shifting. They start to look different, right? And we've even seen individuals who are symptom-free and start to have some movement in their community. So we're very interested in understanding how these communities are changing preceding exacerbation. Are there signals that we can detect that may precede or presage exacerbation? Now, it's also important to look at activity. So it's not just the way these communities are structured, but it's the way they're behaving. And there are ways to do this. And I just want to point out one example here real quickly. This is from Robert Quinn at UCSD. And this goes back to a really old method that some of you may remember from high school biology. This is based on Winogradsky, Sergei Winogradsky. What Winogradsky did was he studied sediments in ponds. And he had this tube, and he would put this tube in the sediment, and what he then would do is study different organisms that lived at different layers of this tube, because there's nutrient gradient, oxygen gradient. So what Robert Quinn did, and this is working with Forrest Rohrer and Kate Whiteson, took a tube, put some media, and inoculated it with CF sputum. Incubated this for a while at 37 degrees with humidity, and they got an oxygen gradient, and then are able to study the way these bacteria are behaving within this gradient. And here are a picture of some of these tubes, and you can study a number of different phenotypes. You can look at pH and redox and biofilm, gas production. And you can do this using sputum from someone who's clinically stable versus someone who's having an exacerbation. And then you can start to map out what the community is doing. You can get these readouts. So here's just two examples. Here's pH, and here's gas production. And this is around the time of an exacerbation. And what you can see here is that with the first dose of antibiotic, with this arrow, you see a marked reduction in gas production, a marked increase in pH. So we're seeing that the community behaves differently at the time of exacerbation than with treatment. So we're now not only looking at structure, we're looking at activity or function of the community. So how is this going to be useful? How is understanding all of this complexity ever going to be really useful? Can we do something worthwhile with this information? Well, the answer, of course, I think, is yes. The more we understand about how these communities are structured, how they're moving over time, how they're moving with exacerbation, how they're behaving in terms of their activity, I think will lead us to better answers about how to manage and understand CF infection. In the shorter term, we may be able to develop some biomarkers. Again, I mentioned if we could see changes that are occurring in the days leading up to an exacerbation, perhaps we could intervene more quickly. Now you may say, how is that ever going to be possible? This sort of analysis takes months to do at this point. How are we going to do DNA sequencing analysis in sort of real time at a patient's bedside? Well, here's how. Technology is moving quickly. Technology will always almost outpace the questions we have. This is third generation sequencing, or single-stranded sequencing. This is a DNA sequencer. Right? This is from a colleague of mine in the UK, in Glasgow, who said he would give me a hand with his presentation. I'll wait. Okay. This is a sequencer. You can put some sample into this, plug it into your computer, and get a readout of the DNA sequence. We are not far from point-of-care handheld devices for doing this sort of analysis. All right, so let me summarize here. Past, present, and future. So pre-Pseudomonas era, development, aggressive use of new antibiotics, marked increase in survival. We now have adolescents with CF, a shift from staph to Pseudomonas, development of social programs, the beginnings of some concern about resistance, to the Pseudomonas era, as I would call it, intense focus on Pseudomonas, a lot of information about epidemiology, outcomes, virulence, biology, antibiotic treatments based on Pseudomonas, not based usually on much else, awareness of interpatient spread now, which then dismantles the social programs that we previously had promoted, decreasing concern about resistance and less reliance on susceptibility testing, to now what I think we are currently in, the Pseudomonas 2.0. Pseudomonas is still very, very important, but the advances in taxonomy, identification of new opportunists, application of these culture-independent methods, important insights into microbial interactions, beginnings of systems biology. This is all where we're going now with CF microbiology. Now, I would be totally remiss, particularly after last night's presentation, to not say anything about correctors and potentiators. What effect will potentiators and correctors have on CF microbiology? I heard sitting at a bar last night that people were saying, well, microbiology is passé. It's gone. Well, I don't know. I think we don't know. What effect will these new therapies have on CF microbiology? When we're able to use these in very young infants, will we prevent infection entirely? What will these correctors, potentiators do to people with already damaged lungs that are susceptible to infection? I think microbiology will still be with us. As we change CF, we will continue to sort of guess. And by that I mean our management of CF continues to change CF. And when I think about this concept, I'm often reminded of this person. Anyone? It's not Sean Penn. I'll give you a hint. Okay? Yeah. This is Werner Heisenberg. And I think about Heisenberg sometimes because I think about the Heisenberg principle. You remember this from high school. And that is, this is usually applied to quantum mechanics, but the principle here is that the act of observing a phenomenon, the act of just looking at something changes that thing. It changes quantum mechanics. And I think about this sometimes with CF. We have uncertainty about what the future changes in CF will look like. But as we look at CF, as we intervene, we change CF. We will continue to change CF with potentiators and correctors, etc. Will that mean we can do away with microbiology? I don't know. I hope so. The thing that I am certain about is that it's very likely that the changes will continue to be very good for people with CF. That I think we can all be assured of. I want to end with thanks to the people who I mentioned at the beginning who provided some really good input into this talk. A number of people provided some unpublished data. I mentioned most of it. I didn't mention that provided by Brian Coburn, David Huang, and David Gutman from Children's Sick Kids, that is, in Toronto, who have some really interesting studies using deep sequencing that'll come out soon. I also want to thank Sam Yanasser and Dick Simon at U of M, my colleagues, always generous with their time and more inclined to treat me like a pulmonologist than an infectious disease doc, for which I'm very grateful. And let me end with my funding, and thank you very much.